Imagine, just scrolling through the news and you see this headline that claims scientists have found, like, a gene linked to schizophrenia. And it sounds like, a really big deal, a huge breakthrough, right? And you see this kind of research pop up all the time, but there's this hidden catch in a lot of brain studies. It's something that most people don't realize.
Yeah, the catch is that this research very often relies on studying brains that are, well, no longer alive, what we call postmortem brains.
That's right. And it might seem like, a really reliable way to study the brain, right? But as it turns out, what we see in a brain after death, well, it isn't always the full picture. It's not that simple. So today, on this deep dive, we are diving into this surprisingly complex world of the postmortem brain, trying to understand how it changes after death and what that actually means for, research on conditions like schizophrenia or Alzheimer's. We're going to be taking a close look at this research paper called, and get ready for it, Selective Time-Dependent Changes in Activity and Cell-Specific Gene Expression in Human Postmortem Brain. It's a bit of a mouthful, I know. But this paper, it reveals some, well, pretty remarkable things about what happens to, our genes after we die. And more importantly, it's going to hopefully give you the knowledge to think critically about those really, attention-grabbing brain studies that we always see in the news.
Yeah, it's about asking those really, crucial questions. How is the research conducted? What are the limitations of the data? Because not everything is always as it seems, especially when it comes to, the brain.
Right. Okay, so to kick things off, this particular study, they did something pretty unique. They compared fresh brain tissue that was taken from epilepsy surgeries to post-mortem samples. So think about that for a second. It's kind of like comparing, a live performance to a recording.
And much like a recording just can't capture every single nuance of a live performance, post-mortem brains just don't tell the whole story of what was happening when the brain was actually still alive.
That makes total sense. But what's the difference? What are we actually missing when we do that?
Well, one of the first things that stood out was the sheer diversity of gene activity in those fresh brain samples. You see, our genes, they provide the instructions for, building proteins, the real workhorses of our cells. But here's the thing, a single gene can have multiple different recipes leading to different protein variations, each with its own specific function.
So it's not just like one gene, one job. It's more like one gene, many possibilities.
Exactly. Imagine like a single recipe, right?
Yeah.
But you can tweak it to create a whole variety of dishes from, sweet to savory. That's kind of what's happening with our genes.
Okay, that's a really helpful way to think about it. But what happens to this complexity after death? I mean, do those recipes just, disappear?
It's not that simple. And that's what makes this research, so fascinating. What they found is that after death, it's not a case of everything just fading away at the same rate.
Yeah.
It's much more selective than that. Certain genes, especially those related to, the rapid fire communication of neurons, those are the first to fade.
So it's like the brain's most, active players, they're the first ones to leave the stage.
In a way, yes. And to make matters even more, well, complicated, the researchers, they discovered that this fade out, it isn't a random process at all. It's incredibly specific. For example, they looked at this gene called RBFOX1, which plays a vital role in, how our brains actually function. In the fresh samples, they saw this beautiful array of, different RBFOX1 recipes, right?
Yeah.
Indicating a lot of activity. But in the postmortem samples, the picture was, well, much more limited.
So it's not just that the overall signal gets weaker after death. It's that, some of the instructions they're disappearing altogether.
Precisely. It's like some of the pages are being ripped out of the recipe book, making it much harder to understand what was being, cooked up when the brain was still alive.
And that has huge implications for, how we interpret research, doesn't it?
Absolutely. Because if we're only looking at the postmortem brain, we might be missing crucial information about what was actually happening when the person was alive.
So how quickly do these changes actually start to happen after death? Are we talking hours? Days?
We're talking about changes that begin within hours. To really understand this timeline, the researchers simulated a sort of, postmortem time machine in the lab.
A postmortem time machine. Now that sounds intriguing. Tell me more.
Well, they took fresh brain tissue and then, tracked how it changed over time, in a controlled environment,
So they basically, fast-forwarded the postmortem process to see what happens, but under a microscope.
Exactly. And what they discovered is that, it's not just this general deterioration of the, genetic material, imagine a book slowly decaying in a library. It's not like that. It's much more, well, targeted than that.
Okay, so it's not about the whole book falling apart, but maybe like certain pages going missing.
Precisely. And the genes that seem to be, most vulnerable are those that are highly, like, active in neurons, the cells that are responsible for, transmitting information in the brain.
So it's like those really busy, always-on neurons are the first to kind of go quiet after death, and their genetic activity it fades really quickly too.
Yeah, it's almost as if the brain, it shifts into this different mode after death, where, some cells actually become more active while others kind of quiet down.
Yeah. That's really interesting. It's like the brain's own like cleanup crew kicks into overdrive.
That's a great way to put it.
Huh.
While those neurons fade, another type of cell called glial cells, they become more prominent.
Glial cells. Now, those are the ones that like act like the brain's support system, right? Providing nutrients, cleaning up waste, that sort of thing.
Exactly. They're essential for just maintaining a healthy brain. And it seems like their activity, it actually increases after death.
So we have this kind of like cellular changing of the guard happening with like glial cells taking center stage as the neurons kind of fade away.
Precisely. And this is where things get, really tricky for researchers who are studying post-mortem brains.
Because if they're like looking at gene activity levels, they might see a very different picture depending on how long after death the brain was, preserved.
Exactly. And that brings us to another layer of complexity in this whole like post-mortem puzzle. The housekeeping genes.
Housekeeping genes. What are those?
So housekeeping genes, they're like the essential workers in every cell. They're the ones that keep things running smoothly. Things like repairing DNA or providing energy.
So they're like always active regardless of what's going on in the cell. Like the electricity in a house, it's always like running in the background.
Exactly. And because these housekeeping genes are involved in, such fundamental processes, researchers, they often use them as kind of a baseline for comparison when they're analyzing gene activity.
So if they're looking at like a gene related to, say, memory, they might compare its activity to a housekeeping gene, see if there's like a difference.
Exactly. But here's the thing. This study found that even some of these housekeeping genes, they aren't as stable as we once thought in the postmortem brain.
Wait, so the baseline itself, it might be shifting.
You got it. And that's a major concern because the very foundation of our measurements is unreliable. Well, it can throw off the entire interpretation of the data.
It's like trying to measure the height of a tree while standing on a platform that's slowly sinking. Your measurements are going to be way off.
That's a great analogy. And it highlights just how cautious we need to be when we're interpreting, research that's based on postmortem brains.
This is all like incredibly fascinating, but I'm sure some of our listeners right now are wondering like, okay, how does this impact me? Why should I care about what happens in a brain after someone dies? It's a good question, and it might seem like something that, only scientists need to kind of worry about, but it really does affect all of us, right? Because when we see these headlines about, some new breakthrough in like, brain research, we need to be able to understand, I think, where that information came from and what the limitations of it might actually be.
Exactly. It's like, reading the nutrition label on a food product. You want to know what you're consuming, right?
Exactly. So when you see a study and it's based on like postmortem brain tissue, don't just take it at face value, right?
Yeah.
Ask yourself, how long after death was the brain tissue analyzed? Was it,
That's the key question. Yeah. Because the longer that, interval is, the more likely it is that some of those like really crucial gene activity patterns, they've shifted.
Yeah.
Like that, recipe book we were talking about earlier with like pages getting all mixed up or even lost.
Right, and it's especially important to be aware of this, I think, when we're reading about research on like really complex disorders, right? Like Alzheimer's or depression.
Absolutely, yeah. Because those conditions, they involve these really intricate networks of brain activity and, gene expression and these postmortem changes that we've been talking about. Well, they can make those networks even harder to decipher.
It's like trying to solve a puzzle, where some of the pieces have like changed shape.
Yeah.
It's still possible to get an idea of, what the bigger picture is, but it just requires like a lot more kind of careful analysis.
Precisely. And, that's not to say that, all research that's based on postmortem brains is, flawed or anything like that. Not at all. It's just that, we need to interpret the findings with a, critical eye. taking into account these postmortem changes.
Right. It's about understanding the nuances, right? The shades of gray, rather than like jumping to conclusions based on some, simplified headline.
Exactly. And this new understanding of, how brains change after death, it isn't about like dismissing past research or anything like that. It's about, refining our approach and making it, even more rigorous.
Yeah, so it's like we're adding this like new lens to our scientific toolbox,
Right.
a way to see the postmortem brain with, greater clarity and accuracy.
Precisely. And that's what makes this field, so exciting. It's this constant, process of discovery and refinement, pushing the, boundaries of our understanding.
Yeah, and this research also raises this really, I think, intriguing question for me. It's something that like gets at the heart of what it means to study the brain, really.
And what's that?
Well, if we can develop, ways to account for these postmortem changes, right? To essentially like rewind the clock on those gene activity patterns, what other secrets of the brain might we unlock?
It's a fascinating question. What if by like understanding the process of death, we gain even like deeper insights into the process of life?
Yeah, it's a pretty mind-blowing thought, right? That like those echoes of life, even in death, could like hold the key to unlocking some of the brain's like greatest mysteries. And that's what makes this whole field so, captivating. It's this constant exploration of the unknown.
Absolutely. And the more we learn, the more we realize how much more there is to discover, right?
Exactly. So the next time that you encounter some groundbreaking study about the brain, just remember that, it's just one piece of a much larger and, constantly evolving puzzle. Thanks for joining us on this, deep dive into the complexities of the postmortem brain. It's a topic that really makes you think, doesn't it?
It certainly does.
Until next time, keep asking those, curious questions and never stop exploring the wonders of science.